So we've discussed this role that solar radiation plays in determining the amount of melanin that's produced in individuals' melanocytes and determining the size of those melanin granules. But when we look at our kind of overall development, we've got to recognize that some of the phenotypic characteristics that we express have evolved as responses to other kinds of environmental stressors. And so I will talk about the response to heat and cold. We'll also talk about the response to high altitude, which is often associated with a hypoxic environment. So, the earliest hominins evolved in the warm to hot woodlands and savannas of East Africa. Therefore, because we all have our evolutionary basis in these earliest hominins, we've got deep seated long term adaptations to heat that are more effective than our adaptations to cold. And so, when you think about what you're comfortable with and what kind of habitat you prefer, and would you rather live in an area that's got a longer kind of hot season or an area that has a longer cold season? Most of us have an easier time coping with heat than cold. I will say I kind of prefer cold myself, but I'm an anomaly, and my kids, even right now, I'll leave the door open at night when it's not all that warm at night, and they wake up and they're freezing and they complain about how cold they are, and they're out bundled in multiple blankets and shivering, whereas I'm like comfortably wearing shorts and a tank top. So I mean, there are some individual differences that deviate from this preference for hot, but in general humans have um, very effective adaptations to hot temperatures, less effective adaptations to cold temperatures. So we have, for example, sweat glands, and we all have roughly an equivalent number of sweat glands. It's about 1.6 million, I think, was the estimate that your text has. There are populations, though, that don't effectively sweat. I have a colleague who is an archaeologist, and her father is of Asian descent, and she can't do, for example, desert southwestern archaeology. She has to do work in tropical rainforests because she has a reduced capacity to sweat. So, Asian populations where it's typically been colder have less effective sweat response and do better in kind of humid environments. So, when we think about the evaporative cooling role of sweat and such, we can think about the difference between wet heat and dry heat. Um, when I first moved out to New Mexico from Virginia, Virginia is really incredibly humid. So, the one of the comments that everybody said was, "Oh well, out there you're going to have a dry heat," and I've grown to appreciate that statement. My mom is like, "Hot's hot. If it's 117 degrees, it doesn't matter whether it's wet or dry." But I mean, at least in a dry heat, if there's a breeze or if you go sit in the shade, it's active. It's actually like you feel a drop in temperature. You feel more comfortable. You feel cooler. Whereas in really humid areas, if there's a breeze, it's often a hot breeze. And if you go and sit in the shade because of the relative humidity, you're still just as hot as you would have been um, out in the sun. So, this is again characteristic of this arid environment that we evolved in. That as we moved out into the savanna with increased aridity, we really focused on this evaporative cooling effect of our sweat glands in our skin. We also, in warm climates and warm situations, have what we call vasodilation. This is where the capillaries near the skin surface widen to increase the blood flow to the skin, especially the face. This allows for our increased sweat response, which then makes us more effective evaporative coolers. This also results in our faces flushing. Some people have really strong vasodilation responses to alcohol. I am one of those people. You will know if I've had a drink, even if it's only one drink, because my face will get bright red. And so, I mean, this is indicative of that kind of super active vasodilation. there's a role of your body size and your limb proportions in particular on keeping yourself either warmer or cooler. There are two rules in biology that apply really across mammals at least, called Bergmann's rule and Allen's rule. Bergmann's rule concerns the relationship of body mass or volume to surface area. So what we do is we see larger body size in colder climates because that reduces the surface area through which heat can be lost. Whereas in hot climates where you need a really effective heat transfer, then then you'll have long, lanky body proportions so that you've got a greater surface area with which to dissipate heat. Allen's rule concerns the shape of the body, particularly our appendages, so looking at our limb proportions, and states that you've got shorter limbs in colder climates, again, because it reduces the surface area available to lose heat, longer limbs in hotter climates because it increases the surface area through which to dissipate heat. So we can look at the stature of populations like the Maasai in Kenya, who are agropastoralists in this kind of high UV exposure, really hot desert mixed woodland environment. To those of the Inuit who, you've still got high UV radiation, but you've got very, very cold climates. The Maasai are among some of the tallest people in the world. Average adult male height's around 6'4". The Inuit, the average adult male height is around 5'2" or 5'4", and they are considered short and stocky, whereas the Maasai are long and lanky. So, you see these differences readily apparent just through looking at their body shape. Our body shape is well suited to the environment in which we historically and evolutionarily resided. If we look as we talk about high altitude, for example, we'll find that many Highlanders have what are called barrel chests. These barrel chests serve to increase the volume of oxygen that you can take in by increasing the volume of air that your lungs can breathe in. So we've got multiple selection pressures that have resulted in different body shapes, at least historically or evolutionarily. Now, with our higher rates of genetic admixture, we don't see this match up as perfectly to the environment as we may have once had. We have physiological responses to cold as well, and these combine factors that generate body heat and factors that enhance heat retention. The latter is much more metabolically efficient. Think about the amount of energy you're going to put into your home, maintaining a temperature with good insulation versus having all your windows open and running the heat on high to try to warm up your home, right? Your heating bill is going to be higher if you don't have good insulation because it's harder to heat things up to a temperature than it is to maintain that temperature once you've reached that temperature. So we have an increase in our metabolic rate and shivering as short-term responses. Both of these generate body heat. For example, if you've ever done a lot of outdoor activities during colder climates like skiing, snowboarding, even snowshoeing and hiking, versus doing those, mountain biking, because you wouldn't really ski in warm weather. Doing things in warmer temperatures, you're going to see a difference in both your hunger and thirst during cold times relative to warm times. In warm times, you're going to be more thirsty because of your sweat and water loss through that mechanism. In cold times, you're going to find that you're really, really hungry because your metabolism is having to work harder to keep you warm. We see vasoconstriction as a short-term response that restricts heat loss and conserves energy. This is where the capillaries constrict, kind of moving heat away from your extremities and keeping it in the core of your body. This is really, really interesting in some bird populations. Birds that regularly, like waterfowl in really cold areas, have countercurrent blood flow, where the arteries that are bringing warm blood to the extremities go right beside the veins that are taking cold blood back to the body, and there's heat transfer so that the warmth stays in the core of the body, and their extremities, their feet and such, are several degrees colder than the rest of their core body temperature. What this does is, and particularly the ability to allow their extremities to be colder temperatures, it reduces their metabolic burden, right? They're losing less heat through their feet that may be paddling in really cold lakes or streams. So we see this in like ducks and swans and geese and such. In general, people that are exposed to chronic cold maintain higher metabolic rates than those that live in warmer climates. The Inuit, for example, have a metabolism that's 13 to 45% higher than non-Inuit, depending on which Inuit you're working with and which non-Inuit populations you're working with. They eat a diet, though, or have traditionally eaten a diet that allows for this difference in metabolism. I mean, the diet's high in marine mammals in particular, with their the blubber, the fat layer that they deposit under their skin. Those are really high fat, high protein diets, which are rich enough to support this higher metabolism. There are long-term responses to chronic exposure to cold. In areas where frostbite is not a risk, there's continuous vasoconstriction. This would include like Australian Aborigines that sleep outside, uncovered and unclothed for much of the year. Those temperatures can get close to freezing at night, but not drop all the way to freezing. And so the Australian Aborigines keep their body temperature, or at least their extremity temperature, a few degrees lower than other parts of their body in areas where frostbite is a risk. So, for example, among the Inuit, there's alternating periods of constriction with vasodilation, so that they will constantly kind of warm up and their cheeks will flush and such, constantly reheating the extremities that during other periods of time are maintained at colder temperatures. So this, by heating them up, it allows you to prevent this frostbite setting in, which can, of course, lead to loss of parts of your body. One other way that we can keep ourselves warm in this context of long-term exposure to cold temperatures is through developing a subcutaneous layer of fat. And we all have fat immediately under our skin. It's part of kind of a mammalian response, and in particular, kind of our human response. But we see population level differences based on the temperature of the area in which your ancestors evolved, as well as the temperature in which you yourself grew and developed. So we can see individual level variation in like the thickness of this subcutaneous fat and underlying metabolic rate, as well as population level variation. So high altitude is very well studied in both old and new populations. This is one other example of how changes in the microecology can lead to some pretty big changes or adaptations for these particular isolated kind of conditions. There are numerous environmental challenges at high altitude. One is hypoxia, or insufficient levels of oxygen. It's not because there's absolutely less oxygen in the air. It has more to do with barometric pressure and the oxygen pressure. When the oxygen pressure is really high, it's harder to get that oxygen out of the air. So we also have more intense solar radiation at higher elevation, increased cold, aridity, wind, a reduced nutritional base, predominantly because there's a lot of plants that don't grow as you go up in elevation, and a harsh terrain. High elevation also plays an important role in reproduction. And so for populations living at high altitude, we've got increased infant mortality, we've got increased miscarriage, incidence of low birth weights, premature birth, and an increased likelihood of preeclampsia. That preeclampsia may be as a response to the need to get more oxygen to the developing baby by increasing maternal blood pressure to pump more blood to the developing fetus. there are ways that we acclimate in the short term to high elevation. One of those, we have an increased respiration rate. If you're in Albuquerque and you've gone from, I don't know, the area by the Rio Grande all the way up to the top of the Sandias really quickly. You just drove straight from, I don't know, like the zoo to Sandia Crest, and then you tried to run up the steps to get to the observation area to look down into the city. You're going to hyperventilate. You're going to breathe heavily. That increased respiration rate is trying to bring more oxygen into your lungs. You also end up with an increased heart rate to transport that oxygen through your body. Um, and then on a slightly longer term scale, but still considered short term acclimatization, you increase your red blood cell density. So this is one reason why people train at high elevation. So people particularly who want to do endurance kinds of sports like marathoning or Ironman or century bicycle rides, etc. By training at high elevation, you're tricking your body or your bone marrow to produce more red blood cells per unit volume blood. This gives you more sites where hemoglobin can bind oxygen. So basically, your capillaries in your lungs are flushed with red blood cells so that every bit of oxygen that's breathed in can be bound and effectively brought into your bloodstream and got into your cells. This is the equivalent of legalized blood doping. I mean, one of the methods of blood doping, well, two methods of blood doping. One is to actually take out your own plasma, run it through the centrifuge, and then give yourself an IV treatment just of the red blood cells that you've separated from your plasma. One other method of blood doping is through taking drugs that artificially favor your bone marrow to produce higher density of red blood cells. So both of those are frowned upon in, in the sporting world, but training at high elevation is not. So it's a way to kind of circumvent that. And then you go and you do your races at sea level and you have this competitive advantage. This response takes about 2 to 3 weeks. So the very first day, if you go to an area that's significantly higher elevation, the very first day or even the very first week, you will hyperventilate, you will have a higher heart rate because you're trying to compensate for the lower oxygen concentration. But after several weeks of training at that elevation, then you acclimate. Your red blood cell density increases to the point that you are able to cope with it without having to increase your respiration rate and your heart rate, etc. There is also developmental acclimatization, including growing larger hearts and lungs and having more efficient respiration. One of the things that happens when people grow and develop in high elevation is that there's increased vascularization of your lungs. So that means that there are more blood vessels in your lungs, and then your lungs are also larger. So both of those are geared towards bringing in higher volumes of air and then having those binding sites through your red blood cells with their hemoglobin to catch the oxygen when the oxygen hits your lungs. These are not things that then go away if you move to lower elevation. You're still going to have more efficient respiration because of where you grew up in your development. And then we can have long-term genetic adaptation. This can include alterations to the maternal blood flow to the uterus while pregnant. So if you grew and developed at high elevation, your kind of uterine physiology will be different when you yourself and we see characteristic genetic adaptations that foster this at the population level among people that have certain ancestry. There are different methods of burning glucose to save oxygen. We see this prevalent in both areas like the foothills of the Himalayas as well as the high Andes in South America. There's a mutation for a decreased ability to manipulate your red blood cell density. This is because having too many red blood cells can cause other health problems. And so particularly for individuals who have for long periods of time, their ancestors have resided at high elevation, they're not going to want to modulate their red blood cell density because that's going to be more likely to lead to an adverse response. And then they also have different body shape with a barrel chest. That barrel chest then increases the volume of air that you can take in with each breath. When you couple that with the increased vascularization of the lungs, then you're kind of perfectly poised to both breathe in large quantities of air and thusly oxygen, as well as to bind that oxygen and transmit it to all the cells in your body. One last component, kind of bioculturally, that has affected our evolutionary history is this coevolution with pathogens. When we were living as nomadic hunter-gatherers, pathogens were a lot less important in shaping our evolutionary development. And the reason was, there's a great scene in the film Nye Story of a Kung Woman where Nye herself expresses that when they were sick, they moved away. You wouldn't stay, for example, around a contaminated water source when individuals were getting gastrointestinal illnesses or parasites. If there were a zoonotic disease that broke out from like one of your prey items and people were getting sick, you'd move away. You'd go hunt something different. You also lived in very low population density and you were outside. So if someone sneezed and released pathogens in their sneeze, it's not in a confined space. Those pathogens are able to dissipate with a lot lower likelihood of infecting someone else. So the communicability of diseases was different. That's not to say that that nomadic foragers don't have any prevalence of disease. It's just to say that it may not be quite the strong selective pressure that it is in modern populations. Once we domesticated plants and animals, we see an increase in urbanization. This is then associated with higher mortality. Cities are population sinks, not population sources. Population sources are where your population growth comes from. Population sinks are basically where your populace goes to die. And we can think about the characteristics of early cities. First of all, you have higher population density, which means a greater risk of communicability, right? Because you're going to have people there to sneeze on when you sneeze. You also have sanitation issues. What are you to do with human waste, for example, right? We used to not have indoor plumbing, running water. We'd use a chamber pot at night. What would you do with your chamber pot when you woke up and it was full? You'd have someone dump it out the window for you. There would be people below living in the alley that are now exposed to your fecal contamination just simply through you dumping it in the river. Think about rivers as sources of drinking water, right? In this increasingly urbanized context, rivers get contaminated. Your livestock drink from your rivers just as your people drink from your rivers. That leads to fecal contamination in the water supply. People would go to the bathroom in the river or dump their waste in the river. People would dump dead bodies in the river, etc. All of these contaminate your drinking water. There's one anecdotal incident for me that really struck me while I was in Indonesia that summer of 1999. We were driving and we stopped at a restaurant. That term is used loosely. This was somebody's house where they offered to cook us food for a price. And we needed to use the bathroom before we got back on the bus. And so they allowed us to use their restroom, which was, they were high on the riverbank, kind of above the river. And their restroom consisted of just a hole in the floor. And so there we are going to the bathroom through a hole in the floor, looking down below at the river where there are children playing and women both getting their cooking water, but also then washing clothes. And that was one of the times where I felt like the biggest, pardon my French, but asshole in the world. Because here I am going to the bathroom where it's going to land on children that are down playing in the water. This is more the norm in underdeveloped areas of the world than it is kind of the exception. So that increase in both contamination as well as in communicability. Also with cities, you get vectors for disease. So you get things like high rodent populations. Rodents carry things like fleas, and fleas give us things like the plague. Even in Modern New Mexico, we are an area where plague is endemic, and so you're not supposed to handle dead rodents, for example. If you find dead ground squirrels or even dead rabbits, leave them alone, call the health department because they will want to test those for the plague and administer a safety protocol in the case that those rodents are carriers of the plague. There are also effects of small versus large group size. It's harder for diseases to become endemic in smaller populations because if you're too virulent, you kill off everybody. If you're not virulent enough, their population is so small, everybody develops immunity. So because of this, we see more endemic disease in cities in high population density areas. The domestication of animals led to an increase in zoonotic diseases and parasites. Tuberculosis and influenza are both zoonotic in origin. So, our communal living with animals and livestock in particular has given us both of these diseases. Both of these diseases are possibilities for the quote unquote next great global pandemic. Not great as in oh wow, this will be fun, but great as in the scale of its magnitude. So we are overdue for a global pandemic. The last one was the 1918 Spanish flu. And so influenza is one of those candidates. We're also seeing the emergence of antibiotic resistant tuberculosis. It's already in New Mexico. And there, as of now, are only two antibiotics that can even treat tuberculosis. And these newer strains are resistant to both of those. If you have a positive tuberculosis infection, you have to undergo about 6 to 9 months of antibiotics. If you have tuberculosis exposure, sometimes you have to go through 6 to 9 months of prophylactic antibiotic treatment. So our use of antibiotics is also then fostering these drug-resistant strains of things like tuberculosis. Your text gives you an example of HIV and AIDS. There are increasingly a number of people in areas where HIV is very prevalent who are able to live with the infection without developing full-blown AIDS, and some who've been identified as not being infectable is not really a word, but they're not able to be infected with HIV. They shed the virus as soon as they have exposure. These are relatively recent patterns to emerge in response to an increased disease risk. So over time, we're going to see more and more of the population in areas with high HIV infection, like sub-Saharan Africa. We're going to see more people that are able to shed the virus or not even be infected with the virus. And so we have this continuing impact of infectious disease on our own evolutionary history. We are breeding superbugs, right? We are going to change kind of the function of our immune system. In a modern context, we don't have normal immune onslaughts because we do things like, and this is going to be counter to this last bullet point, we do things like vaccinate, which reduces the exposure to childhood disease and illness, particularly ones that used to be prevalent, which is great in the sense of we can eradicate diseases that typically had really high child mortality. But the flip side of that is that if we don't get out and get dirty and expose ourselves to pathogens, if we don't have normal, reasonable challenges for our immune system, our immune system is going to be less capable of functioning when we're adults because we haven't primed it, and B, our immune system is going to attack itself or our own bodies. And this is part of what is responsible for the rise of autoimmune disorders. Things like rheumatoid arthritis, lupus, multiple sclerosis, even really intense food allergies like the peanut allergy, right? Your immune system doesn't get to target pathogens, so it targets peanut proteins instead. This is problematic, and really these are challenges of they're first world problems. They're not third world problems. So HIV is an example of evolution and adaptation in a pathogen. It arose from a zoonotic source, most likely SIV in chimpanzees in populations that were butchering and hunting chimpanzees. Influenza has rapidly changing strains to the point that we need a new flu vaccine every single year. This rapid evolution of the influenza virus is part of what makes it a likely candidate for a global pandemic. Malaria is driving genetic adaptation through the sickle cell trait, but we are starting to see drug resistant forms of malaria as well. And so we're going to have to constantly, we have what's called an evolutionary arms race with pathogens, where we are constantly having to evolve molecularly to mount a better immune response. It's part of what goes into who we find attractive, particularly attractive through smell, is us picking up on cues of underlying immunocompetence. But the problem with pathogens is they evolve so quickly. So we are essentially racing the Red Queen, right? Running as fast as we can, evolving as quickly as we can just to keep up, not to move ahead of pathogens. Tuberculosis is increasing in prevalence. A third of the world's population is infected with TB. And TB can be deadly, particularly when it's not responsive to drugs. So we tend not to think very much about tuberculosis in the United States. The conditions in which you would have to be tested for tuberculosis are if you work in like zoo populations, because a lot of the zoo animals can also get tuberculosis, or if you work in childcare. Most childcare facilities require proof of negative TB or no active case of TB. But with drug resistance TB, we're going to see a mounting problem. And it's already moving into New Mexico. So this is not something that we only find in like tropical areas. We also have the impacts of population demography. We've got a growing population size and density. We've got global climate change. There are areas of the tundra that are thawing for the first time in millions of years that have the potential, at least, to release pathogens that we have no idea what they are, what their mechanism is, or how to fight them. So, global climate change is responsible for a couple of things. First of all, more places are warm and wet. It's expanding kind of the reach of the tropics, which means we're getting a wider spread of mosquitoes, which means we're getting more mosquito-borne illnesses. We also, in North America at least, are seeing more tick-borne illnesses as well. Global climate change is also releasing pathogens that have been removed from sites, like these ones that have been frozen in the tundra, releasing them back into the world for the first time. So we may not know how to combat all of those. We've also got anti-vaccine movements, and this has been really prevalent in the news in the United States right now because we've had some measles outbreaks and mumps outbreaks. The measles outbreaks are up in Washington State. Now, the vaccine thing, as I was already mentioning, it's kind of, I mean, there are pros and cons, right? Vaccines have eradicated many childhood illnesses, but they have also reduced the onslaughts and activation of our immune systems when we're younger. So there is some evidence that, for example, having measles as a child is associated with a lower risk of cancer as an adult because it primes your immune system. Now, does that make it worth the risks of adverse kind of side effects or secondary infections to measles on a global scale, on a population scale? Probably not. It's better to vaccinate everybody against measles. But, these are some of the things that at least people who classify themselves as anti-vax or reluctant vax, these are some of the things that they're considering in their research. So vaccines are great, particularly for things that have a lot of adverse reactions, illnesses that have a lot of secondary infections. It's been in the news lately that they've come up with a vaccine for the common cold. That's one that I don't necessarily feel like is a good thing, because the main way that we get sick these days is through cold viruses and rhinoviruses and such. And so if we start vaccinating for those, we're going to do two things. One, we're going to foster the rapid evolution of new strains of cold viruses. And two, we're going to eliminate the primary way in which our immune system is challenged and primed during young ages, which is going to lead to an overall increase in the incidence of autoimmune disorders and potentially deadly food allergies and such. So, we got to take some of these advances in vaccine research with caution, at least, in terms of our kind of global overall health. All right, so our last little bit is going to focus on forensic anthropology. In your text, this is only addressed through this physical anthropology in practice. But forensic anthropology is a special subfield of physical anthropology that involves applying skeletal analyses and techniques in archaeology to solving criminal cases. It's not just skeletons. Increasingly, through advances in genomics, we've been able to identify DNA samples and sequence the genomes and compare them against databases or DNA samples of known criminals, etc., to implicate or exonerate people in criminal charges. When human remains or a suspected burial are found, forensic anthropologists are called upon to gather information from the bones and their recovery context to determine who died, how they died, and how long ago they died. So there are jobs for forensic anthropologists in places like the OMI or the Office of the Medical Investigator, on police forces. In pop culture, we've got this depiction of bones. Now, that one's not incredibly accurate. Some of the things that bones infers are not necessarily realistic in the sense of you can't get all that information from actual skeletal material. But people who know how to quote unquote read bones are able to, with a fairly high degree of accuracy, at least determine certain characteristics of the individual. Forensic anthropologists specialize in analyzing hard tissues such as bones. There are also other forensic specialists who do things like firearm and residue and such, chemical properties, physical properties. With their training in archaeology, they're also knowledgeable about excavating buried remains and meticulously recording the evidence, which helps to preserve the context in which those remains were found. The techniques that are used to read skeletons can be applied to skeletons of any ages. You're able to, for example, to look at the stages of growth and development in bones and teeth to determine whether the individual is a child, an adolescent, or an adult. So we can look at things like the growth plates and the closure of the epiphyses. You're going to have a supplemental, your activity this week is kind of a big activity where you are going to work through a forensics lab and try to determine whether some skulls and pelvises and things are male or female, child or adult, or adolescent or adult, so on and so forth. As well as to determine the racial background of those individuals. So we can infer some things from skeletons. The epiphyses are the growth plates, and in the skeletons of children and even adolescents, these epiphyses are not fully fused. In adults, they are fused. We also know that we have a biological kind of pattern of fusion so that certain bones fuse in certain age ranges. So things like the pelvis in women, that's not going to fuse until they're into their early 20s. So we can use key bones to determine the age of the individual. Abnormal changes to the shape, size, and density of bones can indicate disease or trauma. The pelvis can also indicate the sex of the person based on kind of the birth canal opening and the angle of the sacrum. Bones marked by perimortem injuries, such as unhealed fractures, bullet holes, or cuts, can reveal cause of death. This is particularly prevalent in cases where children die, and the autopsy then uncovers unhealed injuries or even healed injuries that didn't heal properly, indicative of a childhood full of physical abuse. The trained anthropologist can also identify skeletal clues of ancestry with certain markers that can be associated with racial and or ethnic background. And then certain activities, diet, and ways of life are reflected in the bones and teeth based on the presence or absence of dental caries, residue that remains on the teeth, the wear patterns on the teeth, repetitive use injuries like the lower back marks that we find, not physical marks, but marks on the spine that we find when women spend a lot of their day grinding grain, hunched over on their bent knees, etc. So we can infer some things about their behavior as well through looking at the skeleton. Bones are typically photographed and x-rayed. They may also undergo CT scanning or be examined with high-powered microscopes. This can provide detailed information about the remains without altering them, while also providing a visual record once you do manipulate or alter the bones. So you can get things like the position at death. You can get evidence of old age like arthritis and osteoporosis. You can get a clear image of these growth plates to determine the age of the specimen, etc. DNA analysis can also be used to help establish identity. This type of testing is used in a modern forensic casework. Particularly when we have known DNA samples of either perpetrators of crime or like missing children or missing individuals. This can be used to help provide closure to families whose relatives or loved ones have disappeared. It's also been used, though, to map modern human migration patterns, and mitochondrial DNA and such have also been used to confirm relationships of very old remains with deceased or living descendants. So we have something in the United States called NAGPRA, which is the Native American Grave Repatriation Act, the Preservation and Repatriation Act, that it can do things like return skeletons that are in natural history museum collections or anthropology museum collections to the descendants of those individuals through comparing the DNA from these ancient skeletons to those of modern populations. Other chemical analyses, like those involving isotopes, can provide information about the age of the bones and the person's diet. The data is all combined and studied to draw conclusions about the deceased individual. For modern cases, a photo of the skull may be superimposed on photos of missing persons to look for consistencies between bone and flesh form. There are also photo reconstructions of skeletons where having an underlying knowledge of kind of where fat pads are in the face and the way that the muscles overlay the bone, helping to flesh out skulls of found individuals to get an idea of what they may have looked like when they were still alive. This can help identify or even crack some of these old cases of missing persons. Even in cases where no photos exist, that's what I was just saying, that we can reconstruct the face based on the underlying bone structure and known standards of facial tissue thickness. So some of what you're going to do then in your additional learning activity for this week is work through a forensic anthropology lab where you're looking at pelvis, you're looking at skulls, you're determining things like ancestry and sex and even age. And so these are the kinds of tools that forensic anthropologists use in their own active research and in this context of criminal investigation. So you get to kind of try it on and decide if this is something that you really, really like before pursuing maybe more classes in forensics. All right, so in summary, the concept of race, we talked about the concept of race based on visible traits. It's simplistic and invalid. It doesn't take into account evolutionary factors and adaptation to environmental conditions. Since the 1990s, the development and rapid application of comparative genomics has rapidly expanded the genetic data that's available, allowing evaluation of human populations through through thousands or hundreds of thousands of precisely defined DNA sequences. This is part of the reason why we say there's more within population variation than there is between population variation, which shows that we don't have discrete biological units known as races. The role of human culture or culture in human biological evolution, or they're applying this biocultural perspective. We talk specifically about malaria and lactase persistence. The role of UV radiation and the conservation of folate in favoring dark skin in tropical areas, and correspondingly the role of vitamin D production in selecting for light skin at higher latitudes. This is what's responsible, at least in the historical distribution of skin color, which also then, of course, was used to justify institutionalized racism and prejudice. The acclimatization of to environmental forces such as heat, cold, and high altitude. The different ways, both long and short term, that we can cope with environmental stressors. How culture has changed the way we interact with infectious diseases, particularly from the dawn of agriculture on, and how that's going to shape even the future direction of human evolution. And then what's not pictured on this summary slide, what forensic anthropology really is, what we're able to infer from bones and skeletons and how that might be useful, both looking at very old populations as well as looking at more modern populations.